In this video, we're going to go through some of the introductory terms, some of the basic parts of muscles, so that we understand all the parts of a muscle, kind of the basics on how they work, and then we'll get into really the nuts and bolts of how muscles can contract. So, always starting out with my PowerPoints, I have some very common prefixes, root words that you're going to see. And again, we're in the muscle unit. There are several prefixes that all mean muscle. Anything that just has my or mys means muscle. Sarco means muscle. So you're going to see these prefixes in a lot of words throughout this PowerPoint. Just know they're all talking about muscle. I'm going to say a couple other ways we'll get to eventually, but just know you're going to see my, mys, and sarco a lot in this particular PowerPoint. Now, muscle tissue itself equates to about nearly half of your entire body mass. And the main job of muscle is to transform chemical energy in ATP, and it's the energy in those bonds, that's that chemical energy, to some type of mechanical energy, such as movement. Now, doing that's going to exert some type of force. Now, we do have three types of muscle tissue found in the body: skeletal, cardiac, and smooth. And again, my note, you're going to see my or myo, mys, and sarco in a lot of these terms this PowerPoint. Now, a couple differences between the different types of muscle, although we will be focusing mostly on skeletal muscle in this introduction. But a couple things about the different muscle tissues themselves. Skeletal muscles, these are the muscles that are attached to bone. Skin is also attached directly to it. They have really long cells called muscle fibers. The cells are so long, we don't even call them muscle cells, we call them muscle fibers. When we look them underneath the microscope, the cells and that muscle tissue looks striped, it's striated. It's voluntary, which means you have conscious control over your muscles contracting. Although some are reflexes, some will move based on reflexes, but you have control over your muscles. They can contract very, very quickly. They can tire really, really quickly. But they are also extremely powerful, that yes, given the right circumstance, a person could lift a car up. They're extremely powerful. They do require our nervous system stimulation, which is why we've already talked about the nervous system. And they are cells that have many nuclei in them. So most cells have one nuclei. Muscle cells, they're so big, they have lots of nuclei. Cardiac muscle, only place we find it in the body is the heart. It's also striated, and I'm like, it can contract without nervous system stimulation. It's in, there's going to be some type of electrical signal, but there are cells in our heart that will cause their own electrical signals without our nervous system telling them to contract. They're involuntary. You can't control how fast your heart beats. You can do things to, speed it up, go run, and I'm like, slow it down, taking deep breaths, but you can't actually control your heart beating. The cells in your cardiac muscle usually have one, sometimes two nuclei, and they have intercalated discs. That really is their small gaps. We'll get more to it when we get into the actual cardiac, unit, but it's so that the cells themselves can transmit information from one cell to the next very, very quickly. And the third type of muscle tissue is smooth muscle. Now, we find this in all of our hollow organs, meaning they're hollow on the inside, but they're usually full of something. The stomach, most of the digestive tract, your urinary bladder, your airways. Main job of having muscle on the outside of those hollow organs is to force fluids or other substances through those channels. That smooth muscle allows that food in your stomach to do a one-way flow through your digestive tract. We find it in the bladder, in the ureters, that urine is a one-way track through those different structures. It's not striated. It's kind of where we get the word smooth from. It's non-striped. It can contract without nervous system stimulation. We don't need our nervous system telling it to contract. It knows how to do it. It's involuntary. You have no control over it. And the cells only have one single nuclei. They're elongated cells, but they're not as long as the other ones. So this is kind of just a nice little summary from your book, just showing the difference. Skeletal, nice striped appearance to it. Your cardiac also has a striped appearance to it. The cells, you're only seeing one small clip of one fiber. The muscle cells themselves run the entire length of the muscle. So when you think of the muscles in your legs or the muscles in your arm, one cell runs the entire length of that muscle, runs the entire length. So you're only seeing a small snippet. Because they're so long, they do have multiple nuclei. They just kind of hang out on the edges of the cells. You can see the little purple things on here are the nuclei. Your skeletal or your cardiac muscle can also have multiple nuclei, usually one, maybe two, that's about it. Those dark lines, those are those intercalated discs that allow one cell to talk to another cell quickly. Smooth muscle, although they are long cells, they look like a cell that's been stretched out, only has one nuclei, and there are no stripes. There's no striations on smooth muscle. Now, a couple other little differences in your book. You can take a look at it in table 9.3. But I'm like, I was going to say, well, it's just showing whether muscle or the nerves controlling the various types of muscles. They don't always have to. Your skeletal muscle can do both aerobic and anaerobic respiration, which means when you're not getting enough oxygen, your skeletal muscles can still contract. You'll end up with lactic acid and they'll be sore. And I'm like, cardiac and smooth, pretty much they're always going to be aerobic. And I'm like, when we're looking at it, or when they're contracting. Now, some special characteristics of muscle tissue. One, they're excitable, which means they can respond to something. They can receive and respond to stimuli, generally some type of nervous system stimuli, some type of ions that are moving around. Those are charged particles. I'm like, but they can receive and respond to stimuli. They can contract. They can shorten forcefully when they're stimulated. No other tissue can shorten when it's stimulated. They have extensibility, which means they're able to be stretched and extend beyond their resting length. You notice this when you have, you just stretch your muscles out, and like you're stretching them beyond their normal resting length. And luckily for us, they also have the characteristic of being elastic, meaning they can recoil back to their resting length. So you can stretch your muscles out beyond their normal resting length, but then they're just going to be, bounce right back. Now, our main functions of our muscles. There's 4 important ones, and then there's some additional ones. One is to move stuff, whether if they're skeletal muscles to move bone, if it's smooth muscle to move fluid, and I'm like, or cardiac muscle moving fluid, but it's to move something. They can help maintain your posture and your body position, your skeletal muscle. They can stabilize your joints, and they can also generate heat. Some other functions, depending on what they are, they can help protect your organs, because your organs are underneath all of them. They can form valves, they can control your pupil size, and they can also cause goosebumps. And so I'm like, because that's found underneath your tissue, those little erector pili muscle. So a couple other additional functions. Today we're mainly going to focus on skeletal muscle. Now, the outside of the muscle itself. One, it has some tissue sheaths found on the outside. There are some cells in there. It helps reinforce the whole muscle itself. But starting on the very outside of the muscle, the very first outer sheath or covering is epimysium. Epi meaning on, MYS, anything that has MYS means muscle, so it literally means a structure on the muscle. So it surrounds the entire muscle itself. The perimysium, I'm just going to zoom to my picture. So the epimysium is on the whole outside. That actually can all join together and become part of the tendon that connects to the bone. The perimysium is the connective tissue around each fascicle. Each of these bundles is wrapped in perimysium. Peri, the prefix means around, so it goes around a bundle. And then endomysium is the innermost one, and we find endomysium around each muscle fiber itself. So around each cell, this is one cell. Yes, there's smaller circles in it, but this whole thing is one cell. We just call it a fiber, and it's the endomysium that covers it. Now, skeletal muscles are going to attach in 2 places. When we call, well, I was going to say one part we call the insertion, one part we call the origin. The insertion is the part that's attached to movable bone. The origin part of the muscle is attached to immovable or the less movable of the bones. The attachments we also are going to call whether they're direct attachments or an indirect attachment. A direct attachment means that epimysium, that outer part, that outer covering of the muscle is going to be fused right to the bone, right to the outer periosteum of the bone tissue itself, or depending where it attaches, it could be attached to the outer part of the cartilage, the perichondrium. If it's an indirect attachment, it means that connective tissue itself, and I'm like, it forms its own little rope-like bundle, and we call it a tendon. Or if it's a big sheet-like, more instead of a rope-like, if it's more of like a sheet-like covering, we call it an aponeurosis. We find that on our skull. We also find aponeurosis on our abdomen. Now, this is just, looking at some of the structures of the muscle, kind of starting on the outside, and we're going to work our way on the inside. We have the epimysium found on the outside. The inside of the muscle has a whole bunch of these fascicles. If we zoom in on the fascicle, inside each one of those fascicles, you have muscle fibers. And then we're going to zoom in on the muscle fiber. Here's the muscle fiber, and the muscle fiber inside of it. So inside of one cell, it has all these myofibrils. And so I'm like, these tiny, smaller parts. Now, muscle fiber versus myofibril, because a lot of students get those two confused. A muscle fiber, we have a set number of muscle fibers. You're never going to gain any more muscle fibers. That's the number of muscle cells you have in your body. Inside of those cells, we have these other kind of tube-like structures called myofibrils. Because everyone's like, well, muscle fiber, doesn't that word mean muscle, fibril? Yes, this is why it gets confusing, because the names almost sound alike. But myofibrils are smaller, and they're inside of the muscle fibers or cells themselves. And the number of myofibrils can change, meaning if you work out and you're trying to bulk up and get nice big muscles, the number of muscle cells you have don't change, but the number of myofibrils inside those cells can change. You can increase those in number, which are going to make your muscle cells appear larger and your muscles will grow. Now, if we zoom in on our myofibril, zooming in on that part, we're going to look at some of these, smaller parts of the myofibrils, something called sarcomeres. It's these repeating units on the myofibrils, and because you can already start to see there's kind of some darker areas and lighter areas, these sarcomeres, and I'm like, are what's going to make our skeletal muscle appear striped. Now, if we zoom in even farther in our skeletal muscle, those dark areas in those sarcomeres is where you have these thick filaments, and then the lighter areas are where you have just the thin filaments, the blue ones, or where you have only the thick but no thin. I'm like, so these kind of dark areas where you have both the red and the blue, and areas where you have just the blue, all this area here is just the blue. It's not as much, it's going to appear lighter in color. Now, we're going to take a look today at the thick filaments, what they are, what it's called, how they work, and the thin filaments, these actin molecules, and how they work as well. Now, the actual skeletal muscle fiber itself, the cell, it's one huge long cylindrical cell. They can be between 10 and 100 micrometers in diameter, so not very wide, but they can be up to 30 centimeters long. That can easily run the entire length of your thigh muscles. They have multiple nuclei all hanging out on the edge. And then a couple terms when we're talking about muscle fibers themselves, the sarcolemma is just another way of saying the plasma membrane. So anything sarco means muscle, lemma means membrane, so it's muscle membrane. It's the same thing as your phospholipid bilayer but we just give it another name. It's the phospholipid bilayer, it's the cell membrane, it's the plasma membrane, we just call it the sarcolemma. The sarcoplasm, is the exact same thing as cytoplasm. It's just it's the cytoplasm inside of a muscle cell, so we give it its own fancy name. It's the only part in the human body where we do this when we talk about different types of cells. We give it its own names. A couple modified structures that we will take a look at in more detail. There are myofibrils. We'll talk about those. There's something called the Sarcoplasmic Reticulum and something called T-tubules that we'll take a look at. But first we're going to talk more about the myofibrils, those smaller little fibers inside our muscle cells. Now, our myofibrils are densely packed. So this is part of one cell. There's your peripheral nuclei. The myofibrils are densely packed rod-like structures that are inside those muscle cells. They make up about 80% of the volume of the cell. I mean, there's still space, there's still other organelles in there. They can contain those repeating sarcomeres, the parts that are actually going to contract to shorten the muscles themselves. And inside those sarcomeres, they contain smaller filaments. They're called myofilaments. But it's on these myofibrils where we start to see those striations, the dark areas, the light areas. And as we'll kind of zoom in, the dark areas are the A band regions. The light areas, kind of the white on here, are light I band regions. And we'll zoom in and talk what's going on in each of those bands. So here's kind of my zoomed in picture of one of these myofibrils. So the A band, the dark areas where you've got both red and blue overlapping. The I bands on either side of the A band, we don't have any of the red, we just have the blue. And so since there's not as much, it's just the lighter area. Now the H zone is the area right in the middle of the sarcomere. It's right in the middle of that thick filament, or right in the middle of that dark A band. The Z disc is the end of a sarcomere. Again, these things repeat. So one sarcomere goes from one Z disc, which is halfway through an I band, to one other Z disc, which is also halfway through an I band. So this whole structure, Z disc to Z disc, is considered one sarcomere. Now the thick filaments, the red ones, they run the entire length of the A band, making it the darker appearance. The thin filaments, the blue parts, run through part of this I band, and they run part way into the A band. The blue does not connect with the blue. There is a gap in there. Now, the sarcomeres, these repeating units, because you can see this is one whole sarcomere, you can start to see, another part of a sarcomere here, another part of a sarcomere here. They align. They just sit there and repeat and line up one after the other, just like boxcars on a train. We've got sarcomeres in a row. They contain they contain the whole A band, with part of this I band and part of that I band. And they're made up of those thick and thin filaments. The skinny little blue, thin, dark red, thick, and it's those two myofilaments, those thick filaments and the thin filaments, is where we're going to be able to cause some contraction happening as how they overlap. So we're going to kind of zoom in. The red, the thick filaments, is a structure called myosin. And this will be on my next slide too, if you want to look ahead at the next slide or flip back and forth between this slide and the next. But the myosin filaments, like the thick filaments, they have, and I'll zoom in a minute, but they've got these cool little heads on them. And again, they run the entire length of the A band. The thin filaments, made up of a protein called actin, is all of the blue. And they don't connect with each other, which allows for some contraction happening. But they run through part of the A band and through part of the I band till they get to this little Z disc area. This is if I was going to make a cross section through some of these different bands, what I would see. An I band on the end is only going to have the thin filaments, the blue. The H zone is going to have is going to have thick filaments only. That happens right here down the M line. I'm going to say the M line, and I should put next to the M line. The M line is where you have thick filaments that are linked all together with different proteins that kind of anchor them all together. And then the outer edge of the A band is where you're going to see both thick and thin. So the outer edges, this is all outer edges, is where you're going to see both thick and thin, both red and blue. Now the myosin, the red one, contained of a protein called myosin, and the structure of it looks like it has two little heads, and the head area is, we call it the business end. And if we look at part of it, because there are two heads and then the tails are woven together, but the heads have two little binding sites. One spot on each head binds to actin, which is our thin filament, and one spot on the head binds to ATP. Again, it's the business end. Stuff's going to happen here. And so the heads of the myosin are going to be able to, one, bind to actin, and two, because we need energy and things to happen, they're also going to bind to some ATP to give us our energy. The thick filament, I should note, because I didn't mention my cross bridges, I'm like, when the heads attach to the actin, I'm like, they cause a link called a cross bridge. So when we say they form cross bridges, it's when they're attached to each other, they form that link or a bridge. Now the thin filament, the blue, also a twisted double strand, another protein called actin. It also has active sites. The myosin heads bind to actin, and they only do that on the active sites. Now, if you notice, I always think these little actin proteins look like little tiny blueberries, and the little dark circle in the middle that's being covered up by orange right now, but the little middle circle on there, that's where the myosin is going to attach. However, in a resting state, there are two substances that are preventing myosin from attaching. Tropomyosin, bigger word, tropomyosin itself, it spirals around the actin. It's this orange. It spirals around the actin. It stabilizes the actin itself. And yes, and I'm like, it's covering all of those active sites. So in a relaxed state, nothing can bind to that. Troponin, those little yellow structures, is a protein that's going to bind to the actin, the big blue molecule, but it's also going to bind to the tropomyosin. And the troponin's really right there just to anchor the tropomyosin in place. It's to help stabilize it. It's to help keep the tropomyosin covering all those active sites. So when our muscles are in a relaxed state, myosin heads can't bind. We're relaxed. We don't want contractions to happen. Now, just looking at these right next to each other, we can see the thick filament, lots of, tails, and I'm all bunched together, but we can see those double heads sticking up everywhere. The thin filament, the actin, and I'm like, we've got that double chain. I still think they look like big, long blueberry strands. And I'm like, twisted around, and yes, all the active sites are being covered up, unless it's in an actual contraction state. Now, thick filament is made up of a protein called myosin. The thin filament is made up of a protein called actin. Kind of, my simpler way of remembering it, which one is made up of which protein. The word thick is a little bit bigger than the word thin. The word myosin is a little bit bigger than the word actin. Not by much, but it works. So the thick filament gets the bigger word, thin filament gets the smaller word. Now, the couple other parts for the structure of these myofibrils that we don't really get to see on the pictures themselves. One is there is an elastic filament. It's made of a protein called titin. It's strong, and it holds the thick filaments, that myosin, in place. It also helps recoil after you stretch. It helps with that elasticity, and it resists excessive stretching, so that when you stretch your muscles, you don't stretch it too much to the point that you could tear any of that muscle. The dystrophin links the thin filaments, the actin filaments, to the proteins on the cell membrane, the sarcolemma. And then in turn, it's anchored to any extracellular matrix. So I'm like, it's another protein, but it just links those thin filaments. Those thin filaments at some point have to attach to something, and they do eventually attach to that plasma membrane, that sarcolemma of the muscle cell itself. Now, another structure inside these muscle cells. So again, we've got that big, huge muscle cell, muscle fiber. We've looked at the individual myofibrils with all their different bands and thin and thick filaments. Mike, but there's another part that we find inside of here, inside of the muscle cells, and it's all this blue. It's called Sarcoplasmic Reticulum. Now, it's just like an endoplasmic reticulum, but again, I get the sarco, it's muscle, and it surrounds all of these myofibrils. So it's going to surround all of these myofibrils inside the muscle cell. Now, as it goes through, most of them run longitudinally. They run lengthwise with the muscle fibers that are the myofibrils themselves, but they do end and they kind of form this little perpendicular cross channel. So you've got lots of them running lengthwise, but you also have some running up and down. Their main job, because the sarcoplasmic reticulum comes up quite a bit, it stores and releases calcium. That's the big thing. It stores and releases calcium. Calcium really is the go signal, that is the go ion that starts contractions and help continue contractions in your muscles. And so the fact that we need that calcium released at points when we're contracting, sarcoplasmic reticulum becomes very important. Another structure is called the T-tubules. It's these white structures going down the middle. Now, the T is, as they go around to each one, they kind of loop up and then they go up into the next muscle fibril or the myofibril itself. They're a continuation of the sarcolemma. That means they're a continuation of the plasma membrane. Phospholipid bilayer, that's in the full outside of the muscle cell itself, does go down. It kind of, invaginates into the muscle, the myofibrils themselves, and it becomes these white structures. So I think it looks white over here. That's because it kind of, goes down inward and becomes these white T-tubules. Now, the inside is continuous with all your extracellular space and fluid. They help increase the muscle fiber's surface area. Instead of just having the surface area on the outside of the muscle cell just here, you have tons of surface area as you have these T-tubules. Oh, here you can see the indent. You have these T-tubules that are wrapping around every single one of these myofibrils in lots of different areas. Now, because they are found right in between where the sarcoplasmic reticulum, the blue, goes up and down, at the end of each Z-disc, because these happen right along the Z-discs, right along the end of the sarcomeres, they form what's known as a triad. Means three. There's three things there. There are the ends of two sarcoplasmic reticulum, so the terminal ends of the sarcoplasmic reticulum, there's two, and then the T-tubule right in between. It forms this little triad structure. Now, the main way muscles contract and shorten, because they don't always shorten, and I'm like, a muscle contraction will always generate some type of force. They don't always shorten, though. And I'm like, my example, if I was going to be doing this in classes, I could hold my arm out and I could put a textbook on my arm. And I could hold that steady. I could hold the textbook in my arm and my muscles not moving. I could have someone continue to add textbooks into my arms. And although the tension on my arms could go up, I'm like, if I'm just holding the books, that doesn't mean my muscle fibers are shortening themselves. The shortening of the muscle fiber only happens when the tension, because you can still increase it, but when the tension that's generated by the myosin and the actin, those cross bridges when they attach, exceeds the forces that oppose it. So I'm like, we're going to put more tension on those particular cross bridges, on the myosin heads attached to the actin, and things that are opposing it. Then we can shorten those muscles. Now, what happens when we shorten it? It's called the sliding filament. We are going to be sliding myosin and actin past each other. Now, this is one sarcomere in a fully relaxed state. Here's an actual picture of one. We've got all these thick filaments, all those heads, and I'm like, and nothing's attached. None of the heads are attached to the blue actin. You can see you've got that nice dark area with all that myosin, the light areas, I'm like, where you've got, just that blue actin that's attached there. Now, during contraction, the myosin heads will grab and they will help move all the actin closer to the middle. That's the M for middle, the M band, the M line. And so as those heads kind of walk along the actin, the actin moves, this actin moves to the middle here, this actin moves to the middle here, and we've now shortened the actin. Whereas it started off way over here, now the end of that the sarcomere is over here. Same thing, as we started way over here, this Z disc was over here, we've shortened the sarcomere, and now it's over here. And so the only thing, the main thing we're doing is just we're shortening or lessening the overlap between these. And so we are just pulling the end of this Z disc or this Z line this way, and we're pulling this end this way. Myosin heads are just going to walk along the actin and allow that to happen. We shorten the sarcomere, and as this sarcomere shortens, the next sarcomere shortens, and the next sarcomere shortens. They're all shortening. They are all happening at the exact same time. It's not one then the next. And I'm like, they all happen at the same time, and your entire muscle itself will shorten. Happens very, very quickly as you are grabbing stuff, moving around all the time. Now, I'll show a couple videos, but the big idea is those cross bridges, those heads, are going to form attachments, and they're going to break, they're going to ratchet themselves back, they're going to grab another actin area, and they're going to just continue moving those actin filaments closer to the middle of the sarcomere, shortening the entire muscle fiber. Again, I'm going to say, bigger picture of it, you've got those myosin heads, they'll actually attach to the actin, they're going to walk along it, and the actin will move towards the middle of the sarcomere. Now, my note, the actin doesn't shorten, the myosin doesn't shorten. The actin is still the exact same length here as it is here. It's just now these two actin filaments are right next to each other. We've lost that area where there wasn't that overlap. Now, the big picture on how to get the entire muscle to contract. Because at some point, we do have to shorten those sarcomeres to, like when we want to shorten the muscles and form these contractions. But it's just not as simple as just myosin heads grabbing onto the actin and moving it closer. There's quite a few things that have to happen. They all happen very, very quickly, but there's quite a few things that have to happen to actually make muscles contract. The one is all the events that happen at the neuromuscular junction, where the end of a neuron is sending electrical information to the actual muscle itself to tell it to contract. What's actually happening, then there's the part where it's, what's actually happening for that muscle fiber to become excited enough to want to contract, because we're going to have electrical impulses. Then we have something called the excitation-contraction coupling, the fact that we've got both that we've excited the muscle electrically, then we're going to have the actual contraction happening, and then what's known as the cross-bridge cycling. How can we continue to have that myosin head moving along the actin? So there's 4 things that have to happen for your skeletal muscle to contract. Now, there's more steps inside of each of those, and we'll break it down and talk about these. But basic steps is, first we're going to talk about all the things that are happening at the neuromuscular junction, where the end of a neuron is sending information. Here's the end of the neuron, sending information to the muscle itself. We'll talk about the things that happen there. We'll talk about how we can electrically excite our muscle fiber. Then once we've excited our muscle fiber, how that action potential can travel inside the muscle fiber itself to cause the contraction to happen, and then how we can keep those myofibrils from continually reaching up and attaching to the actin and moving and shortening that muscle. So I'm like, there's several sub-steps in here, but we'll break each one down and go through them. But we're going to do that in class, because I figured that'd be a little bit easier than trying to explain it all on this intro video. So we're going to leave you right there.